Specific defenses of the immune system, sometimes referred to as the adaptive immune system. The nonspecific, the more technical term, is the innate immune system. The adaptive immune system is really about it's kind of the really heavy hitters of your immune system. They're the more specialized parts of your immune system. They deal with kind of three things. It's recognizing the invader, attacking that invader, and then creating what we call memory cells, so that if that invader comes back, that you're kind of ready to go. And this is kind of the reasons why we even use vaccines, is to really gear up our specific defenses. The specific immune system takes a little bit of time to get going, because it really focuses on the making of specific antibodies, and then those antibodies need to be produced and made. All right, so the immune system itself, like we saw in the video last time, is really just a collection of different tissues. You can't really point to the one place and that's the immune system. Listing here is just a lot of the important pieces of the immune system. So you've got the bone marrow, which really focuses on making the different types of blood cells, the thymus, the lymph nodes, the spleen. Most of the lymph nodes and spleens are really, is kind of where invaders are kind of collected and then analyzed by your body. Tonsils in the back of your throat, another area where, as you swallow food, it's going to rub against your tonsils, hopefully taking any pathogenic material off of that for analysis. This is also why, your tonsils are infected a lot of times. It's kind of the first thing, that does get sick, especially since it's, the back of your throat. A lot of invaders are going to try to get through your nasal cavity, things like cold and flu and things like that. Adenoids, which is another part of your kind of the sinus area. Lymphocytes is the technical term for very specific white blood cells. These are, if you think, site we've talked about before as cell, lympho or lymphatic system. So the immune system is sometimes referred to as the lymphatic system. So these lymphocytes are very specific immune system cells. And there's two types, T cells and B cells. We'll be going through these in the next few slides. So just kind of have a place where you can, write what I just kind of talked about, but I'll highlight some things here. So like I mentioned, your white blood cells are made in the bone marrow, as all blood cells are, and then they travel to different places. If they remain in the bone marrow, that's where we get B cells, and that's why we call them B cells. These are lymphocytes that mature in the bone marrow. Others will travel to the thymus, where they will mature, and these are called T cells, and they have different jobs depending on where they matured. The lymph nodes, like I mentioned, it's like a screening. I always think of like kind of the lymphatic system with which is collecting lymph. We talked about lymph back in the circulatory system. Lymph is any fluid that's lost by your circulatory system during circulation. That lymph travels down lymph vessels to lymph nodes. Nodes are kind of like a security checkpoint, where the fluid passes through the lymph nodes, and that lymph node acts as a filter, and it's going to collect things, and it's going to analyze them and determine if they are an actual pathogen or not. It's here where these pathogens are exposed to white blood cells so that they can learn about them, learn about their proteins, and again, start finding them. This is why your lymph nodes have a tendency to swell early on when you get sick, because they're the ones that are kind of stimulated and get the immune system, the adaptive immune system, rolling. Spleen, just another filter. This one focuses more on filtering it from the blood versus the lymph. The tonsils and adenoids, like I mentioned, that's kind of those in the back of your throat and your sinuses. Um, it's basically another filter to capture different types of pathogens. And the lymphocytes, we already kind of talked about those. So T cells and B cells. T cells mature in the thymus, B cells in the bone marrow. So step one, remember the three-part, the three steps to an adaptive immune response, okay? Recognize, attack, and remember. So let's talk about recognition. Lymphocytes provide very specific defense because they're going to look for very specific foreign invaders. An antigen, we've talked about antigens and we talked about blood typing. An antigen are those proteins that are on the surface of the cell membrane. They're exposed out and they work as communication. Those are like the marker proteins. As we've learned, we talked about this in viruses as well, those antigens have very specific shapes and they signify that it's either supposed to be there or it's not. Okay, so that antigen is any substance that the immune system can recognize. We talked about the A and B antigens on blood types, right, and blood cells. If they're recognized, the immune system doesn't see it as a threat. But if they're not recognized, then it knows that that's not supposed to be there. This also, we talked a little bit about allergies. It's these, it's those antigens that aren't recognized by your immune system. Even something that's not an invader, like something like pollen, is then thought of as a threat, and you get an immune response, which causes allergies. So antigens can be all kinds of things. Pathogens, bacterial toxins, insect venom, people who are allergic to bees, pollen, mold spores, lots of different things can have these antigens on their surface, and they elicit an immune response. Now, once those lymphocytes recognize a specific antigen, they bind to it. Okay, they trigger, and that triggers the immune response. So the immune response is broken down into two major parts. It does and can get more complicated. We're trying to give you kind of the initial here to give you just a general overview. But as you go deeper into biology, you'll learn even more of this. But again, this is just kind of a glaze over so that you have some background. So the immune response is really made up of two parts: a cell-mediated immune response, which is kind of handled by the T cells, and while the other part is the humoral response, which is recognized by the B cells. So the helper T cell, this is just kind of a FYI for the future, so we're going to be talking about this as well. But helper T cells, specifically the CD4 T helper cells, is the white blood cell that is actually attacked by by HIV, human immunodeficiency virus, and that's why, since this is kind of the beginning here, if you wipe out these T helper cells, you don't have any immune response because it's their job to get the ball rolling. So let's talk about the cell-mediated immune response. It's broken down into 5 steps. Macrophages, which we talked about before, that's actually in the innate or the non-specific immune system. So they're just sitting in the tissues. Some are movable, some are immobile, while others are just situated there ready to engulf. They've got those long spindly tentacles. So here comes an invader, the macrophage will engulf it, and then they basically digest it, and they take the protein and they display it. They actually put it out onto their cell membrane so that the T helper cells can then bind to those antigens, which is going to get everything started, right? It recognizes like, oh, this macrophage, ingested an invader. It displays it on the outside, it binds to it, and then it causes that macrophage to release a special type of protein called interleukin-1. Now, interleukin-1, its job is to activate helper T cells, which release interleukin-2, okay? Interleukin-2, what that does, again, these are just proteins or chemical messengers. Interleukin-2 stimulates the helper cells to divide. So now we're going to make more T helper cells, because again, at this point, your innate defenses have kind of failed. They're not able to destroy and completely eradicate whatever this invader is, so it needs help. So this interleukin 2, the helper T cells are going to divide. They're going to divide really fast, and they're going to, it's going to activate the production of another type of T cell known as cytotoxic T cells. Cyto cell, toxic, obviously toxin T cells. These are their job, kind of like the killer cells. Their job is to destroy infected cells and can also attack those cancer cells. They might attack parasites or other foreign tissues. So it's kind of like, you know, the T helper cells come in, they bind to the macrophage, which releases a chemical messenger, which activates the T cells to release interleukin 2, which stimulates the T cells in the area to start to divide fast into these cytotoxic T cells, which then start destroying infected bodies' tissues. Now, that's this one thing which you have to understand is that these step 1 and step 2 are not, it's not like sequential. It's not like step 1 happens, then step 2 happens. This all happens at the same time. And it's all going on while your innate defenses, your non-specific defenses, are also working to fight the invader. Hold on, this is getting cut off here at the bottom. I'll back out here for a second. Let's get this a little bit higher, just in case you can't see that at the bottom. Move that up. There you go. Okay, going back in here. So at the same time, you get your humoral immune response. This is the B cells that are kind of in charge of this. The release of that interleukin 2, that chemical messenger, doesn't just stimulate the T cells, it also stimulates the B cells. B cells have receptors that are complementary. So think like the key and lock that we talked about in viruses, very similar here. Basically, the B cells, there's lots of different B cells around. You're trying to find a match. You're trying to find a B cell that can attach itself to that specific antigen. And when that is found, it turns into what's known as a plasma cell. So the B cell, once the receptors on the B cell connect and fit the key to a lock antigen, it turns into what's called a plasma cell. And the plasma cell's job is just to sit there and churn out antibodies. And these special antibodies have the same shape as the antigen. I should say not the same shape, but the complementary shape as the antigen. So that these Y-shaped proteins, that we call antibodies, can stick to the invaders. This does a couple things. We actually saw it in the video that we watched. I kind of pointed it out, so maybe you can remember back to that when those Y-shaped proteins were like, you know, they were like attracted to almost like a magnet, and they were just sticking to the invaders. But the actual antibodies don't destroy them. They do a couple of things. They basically engulf them, so they'll stick to them on all sides. Which can make them not able to, like, think if it's a virus, if you clog up all of their keys, they can't unlock the doors to get in cells. They also help bind the invaders together. So, like, you might have one antibody stuck on one of the invaders, and then another invader is kind of passing by. It'll get caught on the antibody, and they'll all stick together in a big clump, which makes it even easier when a macrophage from the nonspecifics, parts of the defense, engulf them. Because now they're not just engulfing one at a time, they can engulf thousands of them at a time. So it helps basically inactivate the pathogens, it helps them flag for destruction, like here they are, come get them, and also kind of binds them all together into a big clump. Now, these plasma cells are going to keep doing this. They're going to churn out millions of antibodies, and they're going to go in your bloodstream and travel all around your body. But those antibodies are going to be there for a while. What's kind of cool is that they're not going to last forever. The antibodies aren't going to be there, you know, indefinitely. But some B cells will become memory cells. These are plasma cells that stay plasma cell. And if the actual antigen ever returns, and assuming it hasn't mutated, right? So it has that same protein. So if it's a virus with an antigen on the outside, it has a specific shape. If it doesn't mutate, it's going to come back and have that same shape. You'll never get sick from that again because your B cells will be there. They'll recognize it immediately, start churning out antibodies, which will destroy it before it even has a chance to attack. This again is how vaccines really work. Trying to get those memory cells made before you even get sick, right? So you have a lot of steps you have to get through, the cell-mediated part as well as the humoral part. So you're sick during all that time until your body can get it under control. But if we can get to this last step without you getting sick, then you'll never get sick from that virus in theory, right? So that's the basis behind a vaccine. This just shows kind of the immune response. You can see the kind of two different pathways. This is a macrophage here, right? So here's your antigen. So this is your, whatever, it kind of looks like a bacteria. It brings it in. It then kind of digests it, but analyzes it, and it displays that protein out here. Then here comes a T cell. Here's the specific CD4 helper cell that HIV attacks and destroys. So this is like the step one. This is why you eliminate this cell. None of this can happen, and that's what we call AIDS. Okay, but anyway, so CD4 helper cell comes in, attaches to it, recognizes it. Interleukin-1 is released, which helps CD4 cells start to divide and create an interleukin-2 there. Is going to change them into the cytotoxic T cells. And then these guys just go around and they attach to infected cells because they're displaying that antigen as well, and destroys it, right? Apoptosis. Some of that interleukin-1, these cytokines, those are the interleukins, 1 and 2, sorry, not 1, but 2, stimulates the B cells. The B cells, again, when it finds the correct matching partner, notice that matching partner, it produces these antibodies, these Y-shaped proteins that are released into your body, and these guys stick to the foreign invaders. And they'll stick from all sides. Like one can stick over here and make them kind of get them to a point where they're completely, they can't get into cells. They can't use their own keys and locks, or keys, I should say, to get into locks. So they're useless. Okay, they've been neutralized. So let's talk about the primary versus secondary immune response. Primary immune response, again, it's going to happen right after the exposure. Notice this is antibody concentration. So, you get kind of a jump in antibody, but then it starts to go down, right? As really the immune system is gearing up. And then eventually those plasma cells are made, and you get an explosion of antibody concentration. And this is where you start feeling better, right? So you're sick all the way up to here. At this point, there's going to be a point along here where the antibody numbers are high enough that you start to win the battle. So we've been kind of talking about this, but talking about immunity and vaccination. The ability to resist infection and not get sick can be gained through the primary immune response, right? So that that initial response, and so some people are just naturally immune to certain things, or getting that vaccination. And that vaccine is going to introduce those antigens into the body before you actually have the real thing. And we talked about vaccines already. You've got live vaccines, you've got inactive vaccines, you've got kind of partial vaccines that have partial parts of viruses and bacteria. You've got the RNA vaccines that we talked about. But the point here is that we want to get that antigen in the body to get that the specific immune system going so that it starts to make those plasma cells and memory cells. And we kind of already covered all this, but again, this is the end goal, right? Create those memory cells so that your body is ready to go when the real thing comes along. Some vaccines need boosters for a couple of reasons. Obviously, we want the memory cells to be strong and producing those antibodies on a regular basis, but also there can be mutations like with the flu, which is why you need to get a booster of the flu vaccine every year, so that your immune system really starts recognizing the different mutations, and so forth and so on. So let's talk about some of the things that that can go wrong with the immune system. Basically, these things are due to an overactive immune system, an immune system that might be too sensitive to things that aren't dangerous. And that's what we're going to cover in this part of the slideshow: allergies, asthma, and autoimmune diseases. Got a video for you. Oh, except I can't play it. Why can't I play it? It's not playing. All right, hold on. Let's get it playing here. Dunk. Let's see. Hopefully I've got this video. Yep. Not sure why it's not working, but we'll play it right from here.
What's true in World of Warcraft is also true in your immune system. To defeat your enemy, you have to know your enemy. Uncover its weaknesses. Learn how to see it before it sees you. We've already talked about how your innate defense system keeps out or quietly neutralizes pathogens without too much fuss. But sooner or later, a threat's gonna come along that's stronger than what the first responders can handle. That's when it's time for the adaptive or acquired immune system to step in. While your innate system takes its zero-tolerance policy very seriously and tries to toast any foreign microbe that it encounters, your adaptive system does things differently. It has to be expressly introduced to a specific pathogen and recognize it as a threat before it will attack. As its name suggests, you're not born with a working adaptive immune system. It's slow to act, in part because it takes time for it to shake hands with so many pathogens and get to know them. These introductions may be organic, like touching a dirty faucet in the bathroom or walking into a sneeze cloud, or they may be premeditated, which is why vaccination is pretty much the greatest thing to happen to medicine ever. But once it's been introduced to a potential threat, your adaptive defenses never forget it. And this ability to remember specific pathogens is one of the key differences between the adaptive and innate defenses. Another main difference is that adaptive immunity is systemic. Rather than being restricted to a particular infection in, say, a sinus or a sliced finger, your adaptive system can fight throughout your whole body at once. And it does this by deploying one or both of its separate but cooperating defenses, your humoral immunity and your cellular defenses. Your humoral immunity, which you might not have heard of before, works by dispatching important proteins that I'm sure you have heard of, antibodies. They're made by special white blood cells, and they patrol the body's humors, or fluids like blood and lymph, where they combat viruses and bacteria moving around the interstitial space between your cells. Much of what you know or have heard about or think of when your immune system comes up actually has to do with your humoral immunity. It's why if you had mumps as a kid, you probably don't have to worry about getting it again for the rest of your life. It's also why doctors and nurses and patients who have been infected with the Ebola virus, a disease once thought to be incurable, have lived to tell about it. And it's why vaccinations work. Whether you're protecting yourself from infections or playing an MMO, one of the first steps in any good defensive strategy is to be able to tell your friend from your foe. And in the case of your immune system, that means being able to identify antigens. An antigen could be an invader from the outside world, like a bacterium, virus, or fungus, or it could be a toxin or a diseased cell within your own body. But in any case, antigens are large signaling molecules not normally found in the body, and they act as flags that get the adaptive immune system riled up. So let's say flu virus gets inside of you, and it's floating around trying to find a good good host cell to start multiplying inside of. Before it finds that cell, hopefully it will be paid a visit by one of the stars of your humoral response, a B lymphocyte. Like all blood cells, these guys originate in your bone marrow, but unlike other white blood cells, they also mature in the bone marrow too. And as a B cell matures, it develops the ability to determine friend from foe, developing both immunocompetence, or how to recognize and bind to a particular antigen, as well as self-tolerance, or knowing how to not attack your body's own cells. Once it's fully mature, a B lymphocyte displays at least 10,000 special protein receptors on its surface. These are its membrane-bound antibodies. All B lymphocytes have them, but the cool thing is every individual lymphocyte has its own unique antibodies, each of which is ready to identify and bind to a particular kind of antigen. That means that with all of your B lymphocytes together, it's like having 2 billion keys on your immune system's keychain, each of which can only open one door. So part of your immune system's strategy is just to win with overwhelming odds. The more unique antibodies your lymphocytes have, the more likely it is that one will eventually find, bind to, and mark a particular antigen. Once they've matured, B cells colonize or seed your secondary lymphoid organs like your lymph nodes and start roaming around in your blood and lymph. At this point, they're still naive and untested, and they won't be truly activated until they meet their perfect enemy match. Which brings us back to the flu virus. When the right B cell finally bumps into an antigen it has antibodies for, usually in a lymph node or in the spleen, and recognizes it, it binds to it. This summons the full power of the humoral immune response, and the cell basically goes into berserker mode. Once activated, the B cell starts cloning itself like crazy, quickly producing an army of similar cells, all with the instructions for the exact same antibodies that are designed to fight that one particular antigen. Most of these clones become active fighters or effector cells, but a few become long-lived memory cells that preserve the genetic code for that specific successful antibody. This ensures that if and when the antigen returns, there will be a prepared secondary immune response that's both stronger and faster than the first. This is key to why vaccinations are so brilliant and important, which I'll come back to in a minute. But while the memory cells are just there to hang back and record things, the effector, or plasma cells, are packed with extra amounts of rough endoplasmic reticulum, which acts as an antibody factory. These cells can mass produce the same antibodies over and over for that particular invader, spitting them out into the humor at a rate of around 2,000 antibodies per second for 4 or 5 days until they die. And the antibodies they make work the same way that the membrane-bound ones do. They're just free-floating. So they ride the tides of blood and lymph, binding to all the antigens they can find and marking them for death. Now, antibodies can't really do the killing themselves, but they do have a few moves that could make it hard for intruders to take hold. One of their most effective and common strategies is neutralization, where antibodies physically block the binding sites on viruses or bacterial toxins so they can't hook up to your tissues. And because antibodies have more than one binding site, they can bind to multiple antigens at the same time in a process called agglutination. The resulting clumps can't get around easily, which makes it easier for macrophages to come and gobble them up. And not only that, but while all this is going on, antibodies are also ringing a chemical dinner bell, calling in phagocytes from the innate immune system and special lymphocytes from the adaptive system to destroy these messy little antigen antibody clumps. So the point of all of this in the short term is to keep you healthy, but in the long term, this process also adds to your overall immunity. The humoral response allows your body to achieve immunity by encountering pathogens either randomly or on purpose. Active humoral immunity is what we were just talking about. It's when B cells bump into antigens and start cranking out antibodies. This can occur naturally, like when you catch the flu or get chicken pox or pick up some nasty bacterial infection, or it can happen artificially, particularly through vaccination. Most vaccines are made of a dead or extremely weakened pathogen, and they work on the premise that because a secondary immune response is more intense than a primary response, by introducing a pathogen into your body, you're priming it to fight hard and fast should that antigen show up again. In the case of typically non-fatal infections like the common flu, this immunity should at least spare you from some of the most severe symptoms. But in the case of more serious diseases like polio, smallpox, measles, and whooping cough, vaccinations can be truly life-saving. Now, some antigens, like those for mumps or measles, don't really change much over time, so a few immunizations will leave you set for life. But others, like influenza, are constantly evolving and changing their surface antigens, so immunity to last year's flu probably doesn't work against this year's flu. Still, acquired immunity doesn't have to be active. Babies, for example, naturally obtain passive humoral immunity while still in the womb. They receive ready-made antibodies from their mothers through the placenta and later on through breast milk. And that works pretty well for a few months, but the protection is temporary because passively obtained antibodies don't live long in their new body, and they can't produce effector cells or memory cells, so a baby's own system won't remember an antigen if it gets infected again. You can also acquire this kind of temporary passive immunity artificially by receiving exogenous antibodies from the plasma of an immune donor. This is what recently saved some doctors and nurses who had contracted the Ebola control the virus from infected patients. A serum was made from the blood plasma of other medical workers who had been infected and survived. The antibodies helped defend the patients from the virus before their own active immunity could identify that particular antigen and start creating their own antibodies. It's not the same as a vaccine, which immediately engages your B cells, but it can buy a patient some crucial, life-saving time against an infection that would otherwise quickly kill. But B cells and antibodies are only part of the immunity equation. There are plenty of pathogens that quickly worm their way right inside your cells, where they're safer from the humoral response and free to multiply as much as they'd like. Luckily, your immune system has yet another game plan and a new set of players ready to fight that final battle with cell-to-cell combat. Make sure you catch our final episode next week and learn all about this epic battle royale. But as for today, in our second to last episode, you learned how the adaptive immune...
Okay, so that is the video there. Just kind of one more note before we close this up. That last part about, you know, that plasma, giving plasma to patients. If you remember, that was one of the first things that that we used to combat COVID infections. Was people who had COVID, or got COVID, could donate their plasma. And that would then contain a lot of those pre-made antibodies for COVID, and you could inject that plasma into somebody else who's maybe more prone to the disease and more prone to dying from it, and that would help save lives. So just kind of a callback to that. All right, that is it for kind of the overview of the immune system. We're not, again, that's not everything. You notice that there's a part 3. We're not going to get into that in this class, but this gives you a good insight into kind of how that all works. The rest of the period, you're going to be continuing Contagion, so enjoy.